So this micro lecture is about decompression sickness, and particularly about decompression sickness in diving animals. We don't have the time to go through decompression sickness in extent, but decompression sickness is an important problem for people doing scuba diving. And here you see the results of some when things can go wrong. The problem with decompression sickness you'll see in a second. But basically, remember that during diving, chest compression on descent increases the partial pressure of nitrogen. So in the same way that when you go down, the partial pressure of oxygen increases, it also, the partial pressure of nitrogen increases. And in this case, when that happens, you have a diffusion of nitrogen from the alveoli into the blood, into the tissues. This nitrogen that now all of a sudden has diffused into the tissues, into the blood, is there, has to be going back to the lungs eventually. But the problem is that the release during ascent may not be as fast as it was taken up during going down. If that's the case, you create a situation of nitrogen supersaturation in the tissues, and when nitrogen supersaturation occurs, you have the potential for bubble formation. And these small bubbles that form in the tissue, if they get trapped in the capillary bed, they will prevent or they will occlude blood flow. So you have, this is a situation of a microemboli. Embolization means air in the cardiovascular system, air in the blood vessels that are preventing flow through that blood vessel. So that's the problem. The problem with the decompression sickness is the formation of microbubbles, or even large bubbles, actually in the circulation and in other parts as well. The decompression sickness is not due to nitrogen being toxic. Nitrogen narcosis is another story that happens at depth, and we're not going to talk about this. It's not due to the toxicity of nitrogen, but to the physical solubility because of the pressure. Pressure accentuates the solubilization, the diffusion of nitrogen into the tissues. DCS, decompression sickness, is due to the appearance of nitrogen bubbles in the circulation, particularly if ascent to the surface is too quick. Decompression sickness is particularly important in divers, in human divers, in human scuba divers. Why? Because you dive with scuba gear and you breathe underwater. So if you breathe underwater, you accentuate this problem even more because you are breathing at a higher pressure, so increasing the potential for nitrogen going dissolved into the tissues. For a long time, people defended that decompression sickness cannot happen in diving animals because these diving animals are not breathing down there. We do, and we can get decompression sickness, but diving animals cannot. Essentially, if we break it down, decompression sickness is the appearance of bubbles, one in the circulation, which leads to embolization and tissue ischemia. Means that the tissues that are downstream from these blood vessels with the bubbles will not get blood. Bubbles in the myelin sheath of axons, compromising nerve conduction. Bubbles in muscles and joints, causing intense pain. This is why decompression sickness is called the bends, because decompression sickness is typically seen in the joints. For some reason, there is accumulation of these bubbles in muscles and joints, and they hurt. And this is what would be the typical chart for a scuba diver. Basically, the duration of submersion, for how long you are there, and the depth. Notice that if you are on the surface, you never need to decompress, because you have not actually pressurized anything. But if you go below 10 meters for longer than 2 or 3 hours, then you need the decompression. And if you go below 40 meters for a very short period of time, 20 minutes, you need to decompress. Scuba divers, what they do is that basically nowadays they have computers. Back in my times, we had small tables that we had to carry with us, plastic tables, but nowadays you have a computer that will tell you, now you have to stop. And basically it beeps, and you basically have to stop at a given depth, sometimes 12 meters, sometimes 9 meters, depends on what the computer tells you, and you have to wait there for a period of time, doing nothing. Because if you keep ascending, the risk for this supersaturation of nitrogen, basically putting bubbles in the circulation, increases, right? So the computer tells you, take it easy, stay at this depth now and hang in here for a while, few minutes. The deeper you go, the longer you dive, the longer the decompression steps are. So this DCS, more common in scuba diving, and then you need a stepwise ascent. Treatment can be done in hyperbaric chambers. Say that an accident has happened. Typically, decompression sickness happens when you run out of air, and then you have to go up quick. And then you don't have air. You cannot stop by. That is typical. What do you do? Well, basically, the solution is to put you in a hyperbaric chamber. Hyperbaric chamber, you are put in a dry chamber, but this chamber is pressurized. And the pressurization of the chamber equalizes your pressure at depth. Therefore, when you are put back into pressure, these small bubbles disappear. And then the pressure in the hyperbaric chamber is slowly changed until you reach atmospheric pressure. That's one potential solution. Decompression sickness is particularly accentuated if you do repeated dives. So if you do one dive in the morning, one dive in the afternoon. People that, for example, go to the Red Sea to do diving, right? They want to maximize diving. They do one dive in the morning, one dive in the afternoon, one dive at night. Three dives a day. And they do that for a whole week. What's the danger? Well, the more time that you dive, you have accumulation of nitrogen. So you have to be careful. You have to take that into consideration. What's the main problem that occurs also? People have got the bends not while diving, but when returning home. Because in the airplane, the pressure is even lower. Right? Remember that airplanes run at a cabin pressure around 3,000 meters of altitude. So then it's accentuated. So divers that go onto these diving trips have to consider how many dives you do in the day. And then the day before leaving, perhaps you cannot dive for the whole day, or you can do shorter dives. You have to take that into consideration to avoid these problems. This is mainly a problem for scuba divers. But what do we know for animals? Essentially, these are AMA divers, once upon a time, more modern. Notice that AMA divers now all wear wetsuits. You would think, well, that's good because they are not so cold. Well, what that has done is that now AMA divers dive for longer periods of times because they are more comfortable, they are not as cold. And as a result, now there's appearance of decompression sickness about AMA divers that basically will be diving down to 20 meters, but they do it repeatedly so many times that they can get decompression sickness. Back in the old times, because they were cold, they could not dive so many times. And then it was a way to prevent it that way. How do marine mammals minimize DCS, decompression sickness? Essentially, there is nitrogen uptake in the tissues stops with lung collapse. If you collapse your lungs, there is less potential for nitrogen in a gas form to diffuse into the tissues. Tissue partial pressure of nitrogen typically decreases after lung collapse. So once the lungs have collapsed, there is less partial pressure of nitrogen. And that's a good way to prevent the vents. Even if descending further, the partial pressure of nitrogen no longer increases because of a redistribution of nitrogen in all tissues. So in marine mammals, basically, nitrogen could be redistributed to all the tissues and evened up there. On ascent, tachycardia, that means that basically an increased perfusion of all vascular beds reperfuses vascular beds and dilutes the excess nitrogen. So it seems that in marine mammals, some of these responses should be sufficient to actually deal with decompression sickness. However, not too long ago, Hooker wrote, breath hold diving marine mammals were thought to be relatively immune to decompression sickness, owing to multiple anatomical, physiological, and behavioral adaptations that reduce nitrogen gas loading during dives. However, recent observations have shown that gas bubbles may form and tissue injury may occur in marine mammals under certain circumstances. So basically, something that we thought it was not really feasible in marine mammals could be occurring. And I want to remind you of this slide that I showed you earlier. If you want to look for an animal that does repeated dives, take the elephant seal, right? Look at that. Repeated dives down to 500 meters back up again. Down to 1,000 meters back up again, repeated dives, hardly ever in the surface. How can this animal not have problems with decompression sickness, one could ask? Repeated dives in particular can cause nitrogen supersaturation and bubble formation. We can say that DCS, decompression sickness, has been observed or has been thought of occurring on a few occasions. And I say that the evidence cannot be ignored. This is data from actually some whales, stranded whales. And this is actually animals that were stranded in beaches, some whale species, I don't remember which one it was. And the interesting thing about this is that this coincided with international naval exercises. That was back in 2003. So what happened is that there were some naval exercises in areas where there seemed to be whales that then they were found beached and dead. When they analyzed these samples, what they found is large bubbles formed in the liver, for example. And then these researchers are claiming, well, here we have evidence for decompression sickness, for bubble formation being responsible for the death of these whales. Back in 2003, these results were taken with a little bit of skepticism. People really didn't see, because the way that they actually wrote the paper, they were suggesting that the reason for the embolism had to do with the use of high-intensity sonar. You know that the army uses sonar to locate objects or whatever. So what they were saying is that the impact of the sonar on the whales would sort of, the impact of these sound waves would actually facilitate the formation of bubbles. And the bubbles would be formed by the use of the sonar, which is something that people really did not really understand how it could actually happen. The question is, can exposure, and they were raising this in the paper, but they could not explain it properly. Can exposure to mid-range sonar precipitate bubble formation? The other question was, when these boats are there making sounds using the sonar, they are scaring the whales off, and the whales, instead of doing the regular dives that perhaps they would take a pause and go up slowly, would go to the surface much faster, as another potential explanation for it. Of course, there were some criticisms to this paper, and the criticisms go along with what we have already said, that if the lungs are collapsed, there should not be any decompression sickness, that if the nitrogen was accumulated in the tissues, the liver would be the last place that it would accumulate, and that's where they showed the largest bubbles, the formation in the liver. Why would they accumulate in the liver preferentially? You would expect the bends, the decompression sickness, to happen as it happens in humans, to accumulate in other parts. In humans, as I said, it's preferentially seen in bones, joints, lungs, and even the central nervous system. And they also, authors claim that the histopathological lesions that they actually show would take much longer time to form. This is not a solved issue. This is not a solved issue. Recent analysis, not experimental data, recent analysis suggest that at least it's remotely possible. The idea here is that, of course, you have inert gas uptake. This is the nitrogen that is uptake in different tissues. And then you have decompression and supersaturation. And of course, when that happens, you can have no bubble formation, no problem. Asymptomatic bubbles, what that means is that these are bubbles that are always formed and disappear, resume by themselves. There's no danger either. Spontaneous resolution. But if you have symptomatic bubbles, you can form actually what's the decompression sickness. And then they can be non-fatal, because the animals can recover, or can be fatal, and then it leads to death animals with bubbles. This is a very simple cartoon picture showing how this would potentially be. There's been analysis and models, but we don't have experimental evidence for this actually happen. And that's how I would like to leave it. Thank you very much.